Thank you very much, Peter, for inviting me. And I also thank Emma Smith for helping an old man in moments of distress, I can tell you. It's been good. I went to school in Liverpool. I went to Liverpool University. I qualified in medicine here in 1954. And it was Professor Downey in the Department of Bacteriology, because I'd done quite well, I suppose, in the course in pathology and bacteriology. He suggested to me that I might like to spend a couple of years in his department, and that it might help me when I had to do my national service. And I did. I went into his department and I worked on chickenpox and herpes zoster. Weller in the United States had just isolated the chickenpox virus a year before this, so I was able to grow it, and with immunological methods at that time, I showed that the one virus was from chickenpox was exactly the same as that from zoster. Of course, that had been thought to be the case from epidemiological observations, but it hadn't been shown in the laboratory. So I went into the army, in my national service. I went in with this virological experience, and they regarded me as a virologist. And where did they send me? They sent me to Porton, which many would regard as a stain on their CV, but I brushed the stain away because it was rather useful as far as I was concerned, because it was there that I met David Tyrrell, who was head of the Common Cold Unit, which was situated on the other side of Salisbury. And he suggested to me that when I'd finished my national service, I might like to join the staff at the Common Cold Unit because they had an MRC vacancy there. Well, I didn't say yes straight away, but it seemed like a good idea. It seemed like a good idea. But looking back at the Common Cold Unit and the brief history of what's happened with the Common Cold, in 1930, Doce and colleagues in the United States, they produced colds in chimpanzees. They were able to transfer the infection, but had no success in growing the virus in the laboratory. And in 1942, the American Red Cross and Harvard University set up a base near Salisbury in the UK. And that was set up really to look after troops in Europe during the Second World War if they developed infectious diseases. And after the war, the Americans donated this unit to the Department of Health, and they didn't quite know what to do with it. But it was Sir Christopher Andrews, who was head of the virology at the National Institute for Medical Research. He thought it would be a good place for housing volunteers for studying the common cold. Well, I like Salisbury very much. I mean, I'd visited the cathedral there 60 years before the two Russian spies had a go at it. And of course, it's a magnificent structure, beautiful architecture. You couldn't say the same for the Common Cold Unit on the other side of Salisbury. Nothing beautiful about it, but it certainly served a purpose. And on the far side here, this is where the volunteers were housed. Each one of these is divided into two, so that you could take 12 lots of volunteers at any one time. And the other huts, various ones of them were for laboratories or for housing members of the staff who wished to live at the unit. It was opened in 1946, after the end of the war, and it closed 44 years later. About 20,000 healthy volunteers had taken part. Somebody said to me, well, you know, what on earth did you do? Because I still get colds. Well, I'll come to that in a moment. They stayed mostly in groups of 2 or 3 and usually came for a period of 10 days. And there were also trials on sick patients. I took what David Tyrrell had said quite seriously, and when he whispered in my ear, and by the way, we've just isolated the first common cold virus, I thought, right, I have to go to this place, which I did with my wife Valerie in 1960. Now, of course, at this time, there were all sorts of challenge studies going on, and I was aware of all those that you see here, and some of those that are being talked about today and tomorrow. Please excuse me if I haven't mentioned those, but at my time, this was what was happening, and there was a self-experimentation by Barry Marshall, and we all know what he did. I see people drinking typhoid material. Well, maybe he was the first to actually have a cocktail of feces and trying to determine what Helicobacter pylori did. And he did well. He got the Nobel Prize for that. But there was some serious diseases here, and there must have been quite a few ethical problems. So talking about ethics, and I think I must be careful because I think you know all about this stuff. But if you look at the history books, they start often with Jenner and what he did, and they question whether it was ethical. Well, I think it was ethical. I mean, what he did was far less dangerous than what was going on around him, where people were being scarified and smallpox vesicle fluid was rubbed in the hope that it would produce mild smallpox. Well, sometimes it did, but sometimes it killed the patients. Well, what Jenner did with his background and understanding what cowpox virus would do, I think what he did was ethical. And anyway, it resulted in eventually the eradication of smallpox from the face of the earth by 1980. After that, there was no smallpox cases. Ethical in regard to the Second World War, I mean, the terrible things happened there, as we all know, in Europe and in Germany, and the perpetrators of those medical catastrophes where people had absolutely no say. Of course, they were brought to trial in 1946, and then there was the Nuremberg Code of Ethics that came out after that, and that makes very good, sensible reading. And later on, 1964, the World Congress, Medical Congress, sat in Helsinki and came up with their declaration. And some would say it's a sort of cornerstone of ethical thinking, of ethical do's and don'ts. And every time somebody thought of something, it was revised. So it went through it has gone through 7 revisions. And then there were guidelines issued by various august bodies that we see here, an analysis of ethical issues in 2 big papers. But by and large, I mean, there comes a point when there isn't anything more to say, and it's really a reiteration of what's gone beforehand. But the reiteration is important, I think, because people tend to forget the ethics, at least in my time. And I'll come back to that. Either they just forget or they willfully forget. But there are bad things that have happened. Now, when it comes to the common cold, let me just go through. If you take all everything I've just said, and as a lemon and squeeze it out, what you get are these ethical principles that the volunteers used if it's the only way to answer the question. They should be run by trained investigators, that's obvious. The subject's welfare takes precedence. There should be independent expert review. There should be reasonable and justifiable risks. which clearly must be determined by a body of people and not just by a single person. There should be informed consent, or from surrogates if it's children or people who are mentally deficient. There should be reasonable financial compensation. There shouldn't be huge amounts of money offered so that people want the money and forget all about what they're doing it for. Confidentiality and data security. There should be exclusion and protection of vulnerable populations. And there should be the right to withdraw at any time, even if it's going to mess up the trial. Now, so far as ethics were concerned, in the case of the common cold unit, it wasn't a problem. I mean, people went there and they had drops put up or down their nose, whichever way you look at it, and they'd come. All of them, I imagine, had experienced colds in the past, so they knew what they might be in for. They were examined, and they were told in great detail as to what the experiment was and what they might expect. They gave written informed consent. The only thing asked of them was that they should not violate their isolation, because they were kept separately in the units. Because we didn't want them to mess the experiments up. And they understood that, and I think the vast majority of those 20,000 people who went through obeyed that. They didn't. Occasionally there was somebody who, it was all too much, and they did transgress. And then, of course, they were sent home, never to come back again. As far as my own work was concerned at the unit, I did quite a few things, but let me just point out two papers. One, the distinct serotypes of rhinoviruses isolated from common colds. We looked at 6 viruses, 6 rhinoviruses, and found that they were all different serologically. So we knew then that you didn't talk about the common cold virus as people did, but there were common cold viruses. And secondly, we showed that infection with one virus would protect against reinfection with that virus, but it would not protect against any of the other rhinoviruses that we had at that time. We now know that there are well over 100 rhinoviruses, so the idea that you were easily going to produce a vaccine was well understood. It was not going to be easy. I also looked at parainfluenza virus type 2. It doesn't mention type 2. Types 1 and 3 had been looked at so far as their ability to produce colds, and we showed that the type 2 would produce colds in adults. There were other important studies, for example, on interferon, which was partially successful. I mean, it was hoped that interferon might behave like penicillin to bacteria. I mean, it might kill a lot of different viruses. Well, it was partially successful, but the amount you had to give to be effective was an amount that produced problems more than the cold itself, and that was therefore put to one side. Then there was vitamin C. Now, we've all heard about vitamin C, I'm sure, and Linus Pauling, who won 2 Nobel Prizes. So he was a significant figure, but he had a real bee in his bonnet about vitamin C and said it would cure the common cold. And I think that the Department of Health in this country, because he was such a big noise, they thought this was really true, and they were thinking about putting its use on the National Health Service so that people could take it for preventing the common colds. And it was then that Tyrrell and other people, particularly in Canada, thought we'd better look at this. And to cut a long story short, they showed that whatever amount of vitamin C you gave, it never prevented the cold. It might reduce the duration of a cold by about 10%, but it was, if you're going to have a cold for 7 days, it might reduce it to 6, but it was really insignificant. So the answer is that it doesn't do anything. If you take vitamin C because it makes you happy, well, okay, fine. But if you think it's going to do anything for a cold, you're barking up the wrong tree. Now, I don't know whether everybody knows this, but the Common Cold Centre at Cardiff University was set up 2 years before the Common Cold Unit in Salisbury closed. It was headed by Ron Eccles, and it was devoted to the examination of new treatments for coughs, colds, and hay fever. And it closed in 2017, and more than 100 clinical trials on colds and flu treatments were carried out there. Most modern-day over-the-counter common cold treatments were tested there for their safety and their efficacy. And I'm sure they did good work. They did things that were quite different, as you can see, from what was undertaken at the Common Cold Unit in Salisbury. Now, in 1962, I went to Robert Channock's laboratory for infectious diseases in the United States at the NIH on a sabbatical. I was there for 3 years. Before I arrived, the Eaton agent had been shown to be a mycoplasma. Now, Eaton had isolated what he thought was a virus and shown that it was well associated with atypical pneumonia. But before I arrived, it had been shown to be a mycoplasma, and it had been put into volunteers. It had produced respiratory infections, and it had certainly produced pneumonia in some of these volunteers. They didn't know everything about this mycoplasma, and I was able to help by using some of the immunological techniques that I'd used much earlier on chickenpox and herpes zoster, to show that it was distinct, it was absolutely unique, it was different from any other mycoplasma that existed. And I got, because of that, when they published this paper in 1963 in Science, naming it Mycoplasma pneumoniae, my name you see trailing at the very end there. It was only later, in the next few years, that it became apparent that Mycoplasma pneumoniae didn't restrict itself entirely to the respiratory tract. It did produce other complications, autoimmune disease, cold agglutinins, skin rashes, even arthritis. And I began to realize then that even when you might have a big trial and found out what you thought it did, it wouldn't actually encapsulate everything that organism could do. And I think that's well understood by all of you. But it came to me gradually. Now, Maury Mufson, who was in Channock's laboratory, he'd isolated a mycoplasma called DC63 from an adult with pneumonia, and the question was, did it cause disease? It was found out to be a strain of Mycoplasma hominis. Now, Mycoplasma hominis was the first mycoplasma to be isolated from humans in 1937. And anyway, did this strain cause disease? Well, did it cause the pneumonia? It was given to volunteers, 50 of them, and shown to cause not pneumonia, but pharyngitis with fever and lymphadenopathy. And that was published in this paper in 1965. Now, I did have some part to play in this. Here's a threesome. This is a volunteer at the base of this slide. He happens to be a prisoner in a local prison. That I didn't quite realize that first off, and then I realized, I was thinking to myself, gosh, I'm involved in a sort of unethical trial here. I mean, giving time off for being in prison depends how much time off, of course, and I never really found that, but I always felt a bit funny about it. On the right there, that's Bob Purcell, who became famous later in life because he isolated discovered different hepatitis viruses. And that's me on the left with the torch. I'm the torch bearer. with a shock of black hair, and you can see the ravages of time, what's happened to that. But I did a bit more than just shine the torch, because I was asked to measure antibody. This was a famous laboratory, but the best thing they had, it seemed to me, was complement fixation, and that was too insensitive. If you took a photograph of when I'm shining the torch, what you see on the left-hand side, you can see that there's exudate on the tonsils. And on the right-hand side, that's 19 days later when they're beginning treatment, the exudate has disappeared, and although they've got red, looked to me as though they've got red sore throat still, the treatment results in recovery. Now, I was asked about measuring antibody. Complement fixation, no good. I found that this method of indirect hemagglutination was very good. It was possible to measure antibody, and where are we? You can see that there are 50 volunteers, and we're measuring antibody pre-inoculation. So these are titers of antibody pre-inoculation, very little antibody or quite a lot of antibody. And what you'll notice is that those with very little antibody, those tend to be the ones who develop disease, 80%, whereas those with quite a lot of antibody, a significant smaller proportion develop disease. So there is this correlation, little antibody, a lot of disease, lot of antibody, little disease, and this relationship suggests strongly that this mycoplasma was causing the disease, that is, the pharyngitis. Not pneumonia, but the pharyngitis. Now, at the time I was qualifying in medicine here, Maurice Shepherd in the United States, he'd identified pleuropneumonia-like organisms, which was the old name for mycoplasmas. He'd identified mycoplasmas from Nego men with and without non-gonococcal urethritis. And he said that when he put the specimens on agar medium, they produced very small colonies. And when I say very small, that arrow shows what he's talking about. These are 3 colonies of Mycoplasma hominis, which show the typical fried egg colony, whereas this is really very, very small and hardly seen by many people. And not only that, the organisms that produce these colonies, they died very quickly. So he had a real problem in getting other people to reproduce his work, because if he sent specimens, they often died in the post, and if they got there, they couldn't see the colonies anyway. So he felt that many people thought he was a bit of a fraud. I mean, he told me that personally, but we knew he wasn't, because in Chanuk's lab, Bob Purcell and I, the organisms had been grown in Chanuk's lab and put away in a freezer for a rainy day. And then when the rainy day came, Bob Purcell and I pulled them out, and we showed that the T strains were unique in metabolizing urea. They had a urease, broke the urea down to ammonia, and they would produce a color change in medium, as shown here, from yellow to pink. And these organisms, they were the only ones that would do it. No other mycoplasma did it. So if this happened, you knew that you had a ureaplasma. And it was a much more sensitive method than trying to look for the very, very tiny colonies. In addition to that, if you added antiserum, you could prevent that color change, and therefore you had a serological test for measuring antibody. And with that knowledge, I came back to the UK knowing everything about ureaplasmas that was known at that time, whereas in the UK, nobody knew about them. And when I got back, in pretty quick time, I'd isolated uroplasmas from just about everything that moved, to be honest with you. And I realized then at that time that I was in fact working for the Medical Research Council, and to turn myself into a vet overnight, as you can see here, was not exactly what they might be looking for. And therefore I had to sort of go easy on the animal stuff. And I did, and continued work on humans, and collaborated with people in London like Eric Dunlop at the London Hospital. But we continued to get rather equivocal results. It was very difficult to prove that uroplasmas themselves were the cause of non-gonococcal urethritis. Although occasionally we did have results which suggested that these organisms might be important. If we look at this last study here, we're looking at men in the Antarctic. Now you may think that's a little strange, but men go to a base camp there, and it's a wonderful place for doing microbiological studies because you've got this big freezer. You can just stick, it's -40 or something outside, and so you can just collect specimens and store them quite easily. And I know it seems awful to say this, but men who go there, they are a captive audience, and they're willing to do practically any volunteer studies just to take their minds off the freezing cold. So in one of these studies, a man came into the base camp, and he had urethritis, and he had uroplasmas, and he was treated, and he got rid of the urethritis, and the organisms disappeared, but then it relapsed without further sexual intercourse, and the organisms came back again. And I thought, that's intriguing, and I don't quite know what it means, but it is increasing. It's probably significant, but I didn't think it fulfilled Koch's postulates. Now, what am I talking about? Well, you know what I'm talking about, but let me just reiterate it. Koch was an important microbiologist, German, Robert Koch. He discovered the tubercle bacillus and the anthrax bacillus, and he wanted to find ways of proving that what he'd got was the cause of disease, and he came out with these ideas, or his postulates, that the agent should be found in all, well, he said all to start with, but then he relented a bit and said most sick people, but not in healthy ones. It should be propagated in the laboratory. It should then produce a disease when inoculated into a susceptible animal, and it should then be recovered and grown in pure culture. And he said if you did all those things, then you could say that the organism was the cause of the disease. Well, we have, that was 1884, and 130 years later or so, we should be able to say a bit more. And with further knowledge, we should, in those with the disease, we should be able to say that the organism is detected in larger numbers in those with the disease than in those without, that an antibody response should exist. Well, it may not, you can't say that, but you should at least be looking for an antibody response, and cure with an antibiotic to which the organism is susceptible. That should be possible to show. And I'm not trying to be another Robert Koch. I mean, all this is fairly obvious that so he did what he could at that time, but now 130 years later, one can do a little bit more. And going back to the uroplasmas and whether they did anything, I felt that we hadn't proved Koch's postulates, and that a human challenge experiment was the best option to fill these postulates. Getting volunteers was a challenge in itself. It was easy to go to somebody and say, would you mind having something up your nose? Yes, quite likely. But if you suggest to them that you might have something in your nether regions, no. I found, most people didn't want to know you. And I wondered about the possible risks. There was a risk of developing arthritis, because it was well known that after non-gonococcal urethritis, a proportion of men, quite a small proportion, would develop arthritis. although this was mainly in those who were HLA-B27 positive. Those who were negative, it hardly ever occurred. So you'd want people who were HLA-B27 negative, and it would be a good idea to be cloning the organism so that it was the only organism that existed when you inoculated, and you'd like it to be tetracycline sensitive so you could get rid of it. Well, I couldn't get volunteers, and then one day I was talking to George Sonker, who was head of the department at the Central Middlesex Hospital, and he said, you know, what you say doesn't sound as though it would be a problem. He said, you probably produce very mild disease, if that. I said, well, I don't know. I mean, you've got to do the thing before you can say what it does. He said, I'd do it, you know. I said, you would? He said, yes. He said, you know, if you think about it, there are thousands of people out there. They're doing this experiment just as we stand here. They're doing it for fun. And they have no idea what they're doing. He said, if we did it, and it was that suddenly the we bit that got me, he said, if we did it, it would be absolutely controlled. There'd be no trouble at all. I said, well, you see, I'd always thought about giving it to other people, not giving it to myself. I said, well, we better look at the HLA-B27 positive bit. He said, yeah, we better do that. And I was hoping I'd be HLA-B27 positive. But unfortunately, I wasn't. No, we were both HLA-B27 negative. And I then thought, well, okay, at least I'll be able to increase the number involved by 100%. So we did it. And I know there's an awful lot of stuff on here, but it's really quite fairly simple. This is not all the data. It was published in this, it was in a good journal anyway, Quarterly Journal of Medicine in 1977. And there are 2 strains inoculated. I'm not saying who's who here, by the way, but there are 2 volunteers. This one got this strain of uroplasma from a man with, obviously, with NGU, and this volunteer got a different strain from a different man with NGU. This is the number of days in which these volunteers were looked at. You'll notice that we looked at things in the urine. And we did that for a real reason. And that was because if you stick a swab into somebody and you twist it around, you pull it out, and you keep on doing that day after day, not only does it bring tears to the eyes, but it also will cause a traumatic inflammation. And that's not what you want. You wanted to find out what the uroplasmas might do. So we worked on urine, and that was also helpful quantitatively to quantitate the number of organisms. And here we have the uroplasmas in the urine. We've got leukocytes, and we've got threads, and we've got polymorphs in the urine deposit, and treatment and symptoms. In this individual, there were characteristic symptoms of dysuria, and the organisms were present in the urine, and it doesn't show it quantitatively, but they multiplied 1,000-fold or so in the urethra. And they disappeared when treatment began, and these other signs gradually disappeared after treatment. In this individual, the symptoms were not as great, but there was a lot going on in the urethra, as you can see, and the uroplasmas persisted. They persisted as long as the individual was not treated. Treatment at this point, the organisms disappeared then, and so did most of the other signs, except the threads in the urine. They tended to persist, as you can see, for about 3 months. Now, you'll never find that if you're looking at people who are doing it for fun. It's only in this kind of experiment do you find that sort of thing. What these threads were, they were threads of mucus with polymorphs sort of studied in the mucus. Where they came from, I have no idea, but they were definitely there. And so we said, well, there you go. We proved cost postulates. I mean, these organisms do something, and I think it did have some effect on how people thought about things. Certainly, studies continued, and we now know that there are two species of human uroplasmas, Uroplasma urolyticum, which these two strains happen to belong to, and Uroplasma parvum. Urolyticum does cause more disease, certainly, than Uroplasma parvum. I'm not suggesting in any way that they cause as much disease or severe disease as the gonococcus or chlamydia, but they do something, and they should be not dismissed out of sight. Now, it's clear that I did a lot of work on non-gonococcal urethritis. And sometimes there were men with disease who you couldn't isolate anything from. Nothing, as hard as you tried. And despite that, they responded to an antibiotic. And I thought, well, okay, that could be just by chance. But on the other hand, it could be that there was an organism there that you hadn't discovered, you didn't know about. It was causing disease, it was susceptible to the antibiotic, and that's why you got that result. And I had to go to the United States to a meeting, and I thought, what I'll do, I'll take some specimens with me to Joe Tully's laboratory. Now, Joe Tully, as it's shown here, he had this very sensitive medium for the isolation of spiroplasmas. Spiroplasmas are mycoplasmas which cause disease in insects and plants, and I'm not going to talk about them. But this medium was also very good for the isolation of mycoplasmas. Joe Tully always looked like that. He was working with people in Bordeaux, and he got to know Bordeaux wines extraordinarily well, and so he often had a happy look on his face. And he was really good to work with. And so I took 13 specimens over with me from men with NGU collected at St. Mary's Hospital. I took them in a canister of liquid nitrogen on British Airways. They were put behind the pilot's seat, believe it or not. I mean, things in those days were quite different from, you couldn't possibly do this sort of thing today. And there are stories I could tell you about all this, but for time's sake, I can't. But I got them there, and I quickly put them into Tully's medium. And I was there for about a month, and nothing was happening. The medium contained arginine or urea or glucose. But this day before I was about to leave, and I had to come back, there was a color change in one of the vials, shown here. And of course, it could have been nothing. But I subcultured from this vial to fresh medium, and I came back to the UK, and Joe Tully phoned me and he said, you know, those vials, your subcultures are changing color, and they're not cloudy. It doesn't seem to me as though it's bacterially contaminated. I think there really is something going on, and there was something going on, and it happened to be a new species of mycoplasmas. We found it was a mycoplasma, and we found that it was not the same as any other mycoplasma. It was a new species, and we wrote these two preliminary papers about it, and later we called it Mycoplasma genitalium for obvious reasons. It had a very small genome, the smallest of any mycoplasma known. It grew slowly, as you can guess, from what I've said. It stuck to and invaded host cells. It was inhibited by antibiotics, and it was sort of bottle-shaped, as you see here. Now, if you increase the size of this by about 500,000-fold, you get this sort of 3D effect. And this is Angela Robinson holding Mycoplasma genitalium. This is what one mycoplasma might look to another mycoplasma. She said to me, David, she said, you know, I've never held a genitalium as big as this before. I said, I don't know what you're talking about. But she was a consultant, or she still is, at the University College Hospital in genitourinary medicine. And it's true that when nobody else was able to hold a mycoplasma like this, because nobody could isolate the thing. They couldn't isolate it in the medium they had. And it's very interesting that Joe Tully's medium, when it disappeared, when it was all used up, you could never find medium as sensitive as that again. And neither he nor I, or I could a bit because I got some of this medium back with me in the UK, but we couldn't isolate it again. And it was only later, though, and then the polymerase chain reaction came along, that we could make some progress. But in the interval, I was beginning to think, because so many people were trying and failing, I was beginning to think, you know, they think we're a bit of a fraud. So I thought, well, I'm not going to put it in myself. I've had enough of that game. And I have a feeling from its appearance that it could really be pathogenic, but we could try chimpanzees. Now, I was never going to get chimpanzees in the UK, but from my contacts at NIH, I was able to get 11 chimpanzees. This was, over here, this would have cost about £100,000 or something like that, but I got them for nothing. So I got 11 chimpanzees, and all of them became colonized, but 8 of them were colonized for 5 weeks or longer. That is as long as before they were treated. If they were not treated, then the organisms persisted. And 9 of them had a 4-fold or greater antibody response, and in 7 of them, there was a very good polymorph response in the urethra. This probably did too, but it got bacterially contaminated. So I felt, because you and I are very close to chimpanzees, I felt that we really had fulfilled postulates in this case. And as I said, it was only later that with the PCR, it was possible to show that Mycoplasma genitalium caused all these conditions. And you see in women right through to salpingitis, and if not treated, it would cause infertility. The problem is now that, and looking ahead, that it's showing increasing antibiotic resistance. And you know, the press gets hold of these things, and they love to call it a new super bug. And people tell me that with better antibiotic management, we won't have to worry about it. That is nonsense as far as I'm concerned. I mean, people say, you know, they're astute and they will be able to manage. Well, okay, my colleagues are astute, but not everybody around the world is astute, and they're going to do all the right things to prevent this resistance. And I've said in 1950, what is that, in 2014, 4 years ago, I was writing and saying that you ought to really be thinking about a vaccine. Well, I said it, you know, nobody's taken any notice really. I did say this in sobering thoughts. Somebody said to me, does that mean you're drunk most of the time? No. I mean, if you just think about it coldly and coolly, you come to the conclusion that a vaccine is the thing you need. And I do hope it's going to happen. Now, finally, I'm going to talk about gonorrhea. And when ethics gets thrown out of the window, first of all, John Hunter. I just saw him in the coffee break. He was looking at me from a picture upstairs. Didn't say anything about him being nasty. So I have to be careful what I say, I can tell that. But he was famous, and he mentions it upstairs, for his dissection of bodies. And of course, as you all know, there's the Hunterian Museum in the Royal College of Surgeons showing his dissections. But he also became interested in venereal disease. And he wrote a thesis in 1766, and he said there was only one venereal poison. If it fell on mucosal surface, it caused gonorrhea. On skin, it caused the chancre. So he knew the difference between gonorrhea and syphilis. And in 1767, he tested his theory by inoculating a recipient. Now, there's debate as to whether it was himself or whether it was other people. It's not absolutely clear, but whatever. There was pus from, he inoculated with pus from a patient with gonorrhea. And a sore appeared 10 days later, inguinal gland enlargement. What happened? The individual got syphilis. And this convinced Hunter that he'd induced syphilis by inoculating gonorrheal pus. Thus, the two conditions were caused by one agent, but almost certainly he'd inoculated not only the gonococcus, but he'd also inoculated the treponeme of syphilis. But you couldn't tell him he'd made this mistake because he was a very famous man and he would shout you down. And Hunter's mistake set the field back by at least 50 years. And it was only when he died that people really came out of the woodwork. And one of them was Philippe Ricord in Paris. And he did have morals. He wouldn't inoculate anybody who was healthy. But what he did do was he used this procedure of auto-inoculation. Now, what I mean by that is that supposing somebody came with gonorrhea, he'd take some of the pus and he'd put it under their skin or into their skin. Now, what they said to one another during this process, I have no idea, but that's what he did. And then he'd look to see what happened to the material he'd put into the skin. What I think is really unethical is that he didn't do this just a few times. He did it to 2,500 patients. I mean, you don't need 2,500, surely, to prove your point. Eventually, however, he did come out. He said, if there's a chancre, it only produces a chancre. But if you put gonorrheal pus in, it never produces a chancre. He said, therefore, there are two diseases, and they must be caused by two separate agents. And he was aided and abetted by Albert Neisser. Schoolboy joke is that you'd never meet a nicer chap. Well, this was true. When he was young, he was really nice and he was clever, because just a year after he qualified in medicine, he found the gonococcus. And it was named after him, Neisseria gonorrhoeae, a word devised so that doctors could never spell the thing. It's got 6 vowels in that word. And he was famous, and he often said, you know, where would I be without the gonococcus? Well, he was famous, yes, but then disaster. He got interested in syphilis, and he gave serum patients with early syphilis intravenously to 10 to 24-year-old subjects. Some developed syphilis within a few weeks. There was a public outcry, so he was brought before the Prussian Parliament, who reprimanded him and fined him. Well, I think he got away very lightly. He was outraged and absolutely unrepentant, and many of the people at that time thought, yes, what's he done wrong? I mean, that was the feeling, you know, people could do anything to anybody and think they could get away with it. Thomas Parham, I can't keep on about bad people, but he wasn't good, that's for sure. He was he was Surgeon General of the US Public Health Service from 36 to 48, and he sanctioned the events in Guatemala, which were, to a large extent, kept secret, but you can't keep it totally secret. But there were papers found eventually, which had been secreted away, and it all came out then that 1,300 people had been exposed to gonorrhea, syphilis, or chancroid to find out whether penicillin was effective. And they did a really good controlled study because they treated half of them. but the other half they just left to rot. They didn't do anything with them, and 83 of them died. And it was Barack Obama, as late as 2010, who apologized to the president of Guatemala, saying how sorry, this should never have happened. It was awful. I mean, that is ethics thrown out of the window. He also did something else, which I don't really need to go into. It was just as bad. Now, so far as our own research on the gonococcus was concerned, we were mindful that volunteers should not be used if the aim could be achieved another way. And we were looking at the pathogenicity of different strains of the gonococcus. And we used ovarian tubes. In other words, in the human case, women who were having their tubes taken out, we'd ask them beforehand, and if they agreed, we'd take those tubes and keep them alive in the laboratory as organ cultures, and then add the gonococcus to them. And we found in that procedure, and you must remember that what we're doing is taking the tubes and sort of separating off the immune system. And in those tubes, we found that it was only human and chimpanzee tubes that suffered damage. It was the gonococcus was very specific. We found a soluble exotoxin produced by the gonococcus, which caused damage, and host tumor necrosis factor was produced, which was also responsible for some damage. So we see here that this would be a normal tube with a cilia which beat and pushed the ovum down to the uterus to be fertilized. This on the bottom is the same, but the gonococci have been added 24 hours previously and totally damaged this tube, or soluble exotoxin would do exactly the same thing. So we did have some information about pathogenicity. I'm not saying that you don't get more information from looking at a whole being, because we've taken away the immune system and what that might involve and do. The other thing that we did, we looked at the effect of sex hormones, and we were really surprised when we found that female mice became susceptible to the gonococcus if they had been pre-treated with estradiol. Here I am going on about chimps and humans being the only ones susceptible. Here we had a mouse that was susceptible if you did that to it. And we published that in 1990. I don't think many people in this country took any notice, but in the States, Anne Yersey did. She did a lot of work with this model. She acknowledged what we'd found, and she published a lot. And Plant also published here in 2000, and he showed that the biological feasibility of developing a vaccine using the mouse model. So I think it was useful, and it was useful to the people in the United States who were using humans and what they it helped them a bit, I think, as to what they might be doing. And human volunteer studies have really been the preserve of investigators in the United States. I think the first publication was about 1987, so about, you see the first publication here, 87, so it's about 30 years ago. Here in Hobbs and colleagues talking about the cumulative experience in this publication, and you see that Yersey's involved in that. It's interesting, this is a review, and they talk about the cumulative, and they don't tell you how many volunteers that amounts to, but they do say hundreds of volunteers have participated without severe adverse effects. Well, you have to be very careful, because you can gather that this organism will rush up the urethra and to the epididymis and to the testes, and before you know what, it's causing a lot of damage. But what they do is to treat very early and prevent any of that. So what they've got is really a model of what is happening in the early stages, they don't have it in the late stages. But nevertheless, it is an important model, and I do hope that it's going to lead to a vaccine. 30 years have gone since the first publication, and there's no vaccine as yet, but I do hope it comes, because as you know, in certain parts of the world, the gonococcus is absolutely resistant to any antibiotic that's thrown at it. So on that note, I'll finish. I'll leave you with everything I've said is on that slide. I could acknowledge so many people, thank so many people. I can't do that. I would just say thank you to the MRC, because they supported me and my team for over 30 years, which I think is something. And I would thank one other person. I find this difficult, but I would thank one. You remember what Nisus said? He said, "Where would I be without the gonococcus?" Well, I wouldn't say that, but I would say, where would I be without my wife? I mean, she's been through thick and thin and supported me all the time. And she's here, and I know she's going to be annoyed that I say this, but I do thank her very much. And I thank all of you for being with me on this rather long trip down memory lane. Thank you very much.
Thank you, David. Fantastic lecture. It just reminded me that my first 2 publications as a medical student were self-experiments and done without any ethics committee or signed consent.
Yes.
Another story I just like to relate is a story about vitamin C. I remember being written to by a journalist who said, well, if Linus Pauling says that vitamin C works by drinking it or eating it, how about sniffing it up the nose? So in the spirit of self-experimentation, I ground up some vitamin C and was aspirating it into my nasal passage in my office when Tracy Hustler walked past. And my reputation took a severe dive at that moment, from which I don't think it ever recovered.
Did it do anything for your cold?
It didn't, but it caused terrible pain. So ascorbic acid up the nose is not good. Not good.
One of the best remedies I ever had was cutting an onion in half and rubbing it on your chest twice a day. This was fantastic because nobody would ever want to come near you.
Very good. So, does anyone want to ask any questions, add any reminiscences, or?
You know, Linus Pauling, he took tons of this stuff. He spent his life taking huge amounts. Never realized that, you know, there's only a certain amount that is required. The rest of it is excreted. But he'd take 15 grams of vitamin C a day.
Given the hugely long history of the common cold unit, what was its final death knell? Why was it finally decided to be obsolete?
Why was it finally closed?
Yeah.
I think that the MRC felt that, you know, the business of a vaccine was far beyond it, and that's where things should be heading. And I think they just felt that it wasn't going to come out, and they were spending. You know, it was expensive to keep that unit going. But it had I think it had probably fulfilled its purpose. I mean, it had isolated everything that was important.
Before we go, just one more round.
One more round of applause.